Hi everyone, my name is Vinicius. I am a PhD in ecology at the University of Brasilia, and here I'm going to present to you how fertilization in liming can reduce and displace columbian's isotopic niche, which could be a potential indicator of nutrient input into central Brazilian savannas. The Brazilian savanna, locally known as Cerrado, is a global biodiverse hotspot due to its high levels of above-ground plant and animal species richness, high degree of endemism, and accelerated habitat loss. This is because Cerrado is also an agricultural frontier and had about 46% of its native vegetation converted mainly to pastures for livestock, but also to annual croplands. Together with plowing, liming, fertilization, and exotic grasses cultivation, which hinders the native savanna restoration in abandoned agricultural areas, agricultural activities expand to models that contribute to reactive nitrogen and phosphorus emissions into the atmosphere, which may lead to its consequent deposition into the native savanna remnants. Estimates suggest that nitrogen deposition into Cerrado may more than double by 2050. Outcomes of a long-term experiment of nutrient addition into a woodland savanna have been showing that in addition to promoting lasting changes in soil chemistry, these nutrients can modify plant diversity and cover, promote exotic grass invasion, alter litter quality and decomposition, and affect the soil microbial diversity. All this represents changes in the quantity and quality of resources that support soil food webs and their functions, affecting soil-trophic interactions in ways we do not know yet. To begin understanding whether the legacy of nutrient addition into the savanna may have compromised soil food web interactions, we took a closer look at what columbians are doing. This because columbians are among the most widespread and abundant soil arthropods, inhabiting various organic substrates and using a wide range of food resources. Acting at the base of the food web, columbians contribute to nutrient cycling and channel carbon to high trophic levels. Stable isotope analyses have been showing pronounced trophic niche differentiation among columbian species in temperate and tropical forests, and this differentiation in large has been explained by the taxonomic identity of columbians. Here, by accessing the isotope composition of common epigenic columbians between treatments and normalize the delta 30C and 50N values of columbians by the local literature delta values of each treatment, we applied descriptive metrics based on stable isotopes and found that columbians were accessing less diverse basal resources in NMP treatments compared to control. And as with liming, a large proportion of columbian assemblage members in NMP treatments had similar trophic preferences. It culminated in a reduction in isotopic niche width of the columbian assemblages. And analyze the standard ellipses in the biplot, we also notice the displacement of the isotopic niche of columbian assemblage in N plus P and liming, which may represent the influence of exotic grasses with C4 photosynthetic cycle that invaded this treatment. So our study suggests that increasing nitrogen and phosphorus deposition into native savannas or the liming legacy in restored savanna soils may impact columbian niche structure, and this can have consequences for the ecosystem functioning in ways we do not know yet. But for now, our outcomes bring perspectives on the use of carbon and nitrogen stable isotope metrics on columbians as indicators of nutrient input into savanna ecosystems. And these indicators could help us detect the expected nitrogen deposition increase into savannas and assess whether this savanna restoration in abandoned agriculture areas effectively restores the soil-food web interactions. So thank you very much for your attention and all the best for all of you.